Hello everyone and welcome to a Biol 110 Ambassador Program presentation. Today I'm excited to take you on a fascinating journey into the microscopic world, focusing on mitosis and the compound-like microscope that allows us to explore it. Mitosis is a cornerstone of cellular biology, pivotal for organism growth, tissue repair, and genetic consistency across somatic cells. It's a process where a single cell divides to produce two genetically identical daughter cells, each containing the same number and type of chromosomes as the parent cell. Microscopy is an essential technique in biology that magnifies objects too small to be seen with the naked eye. A compound light microscope, which we extensively used in our labs, consists of several key components, each serving a unique purpose to enhance our view of the microscopic world. The eyepiece magnifies the image produced by the objective lens. Our microscopes typically come with 10 times or 15 times magnification ocular lenses. The resolving nosepiece allows us to switch between different objective lenses, adjusting the magnification power for our specimen. Speaking of objective lenses, these are critical for focusing and magnifying the light coming through the slide. With scanning, low, high-dry, and oil immersion powers, we can observe specimens at various levels of detail. Understanding the magnifying power and how it affects the field of view is crucial. The total magnification is calculated by multiplying the ocular lens power by the objective lens power. As we increase magnification, our field of view decreases, making it imperative to adjust our focus and positioning carefully to observe our target cells. In our mitosis lab, we utilize these principles to not only observe but also estimate cell sizes at different magnification levels. In this segment of our presentation, we will explore a fascinating laboratory experiment that brought the dynamic process of mitosis into clearer view. Our exploration into mitosis was hands-on, utilizing compound light microscopes to observe garlic root tip cells. The choice of garlic root tips was strategic, owing to their high mitotic index, which provided us with ample opportunity to witness the various stages of cell division under the microscope. Our objective was straightforward yet profound, to observe and identify the stages of mitosis in garlic root tip cells. This experiment aimed to bridge theoretical knowledge with practical observation, enhancing our understanding of cellular division. To conduct this experiment, we utilized a compound light microscope prepared slides of garlic root tip stand to highlight chromosomes and various other laboratory tools and reagents necessary for the preparation of these slides. The procedure involved a series of meticulous steps, from preparing the garlic root tips through fixation, staining, and slide preparation to the final observation under the microscope. Each step was crucial for achieving clear visibility of the mitotic stages. Let's embark on a data journey through the stages of mitosis, unraveling the complexity and precision of cellular division. Interphase is subdivided into three main phases: Gap 1, synthesis, and Gap 2. In Gap 1, the cell grows, producing proteins and increasing in size. Then in the S phase, the crucial event of DNA replication occurs. Each chromosome, initially consisting of one chromatid, now becomes a pair of identical sister chromatids, joined at their centromeres. Following DNA synthesis, the cell enters the G2 phase, where further growth occurs, and the cell checks to ensure everything is in order for division. Cellular organelles are duplicated, and the cell assembles the materials necessary for mitosis. It's a phase of preparation and check, ensuring that any damage to the DNA is repaired and that the cell is fully equipped for the next phase of the cycle. Interphase, often overlooked in the spectacle of mitosis, sets the stage for accurate and efficient cell division. Without the diligent preparation during interphase, the cell would not be ready to embark on the remarkable journey of mitosis that ensures growth, repair, and reproduction in multicellular organisms. Prophase, a stage where the cell prepares for division. Here, the DNA, loosely packed within the nucleus, condenses into visible chromosomes. each chromosome, now doubled, is linked at the center by a centromere, appearing as two identical sister chromatids. Meanwhile, the nuclear envelope dissolves, setting the stage for division, and the mitotic spindle, a structure made of microtubules, starts to form at the poles of the cell. Next, we transition to metaphase, a critical point in mitosis. The chromosomes align themselves along the metaphase plate, an imaginary line at the cell's equator. This alignment is crucial, ensuring that each new cell will receive an identical set of chromosomes. The spindle fibers attach to the centromeres of the chromosomes, preparing to pull the sister chromatids apart. Anaphase marks a turning point in mitosis. The centromeres divide and the spindle fibers begin to pull the sister chromatids apart with remarkable precision. As the chromatids move towards opposite poles, the cell itself starts to elongate, making room for the formation of two new nuclei. Our journey concludes with telophase, where the chromatids reach the poles and begin to decondense, transforming back into the tangle of chromatin from which they began. New nuclear membranes form around each set of chromosomes, resulting in the formation of two distinct nuclei. Finally, cytokinesis, the division of the cell's cytoplasm, completes the process, giving rise to two genetically identical daughter cells. Our observations were illuminating. We successfully identified the stages of mitosis within the garlic root cells. This laboratory experiment of mitosis was not just an academic exercise, it was a journey into the heart of cellular biology. It underscored the beauty and the complexity of life at the microscopic level and highlighted the importance of hands-on learning in the scientific process. Let's expand on the real-world connection between mitosis and bioremediation with more specific content and details. This real-world connection not only showcases the practicality of our biological studies, but also highlights how microscopic processes can lead to macroscopic environmental solutions. Bioremediation utilizes living organisms, primarily bacteria, fungi, and plants, to degrade or detoxify pollutants from contaminated sites. The process hinges on the natural metabolic pathways of these organisms, transferring hazardous substances into harmless or less toxic compounds. The success of bioremediation largely depends on the rapid proliferation of these microorganisms, a process underpinned by mitosis. The efficiency of bioremediation efforts is directly correlated with the growth rate of bioremediating organisms. Through mitosis, these organisms can replicate quickly, increasing their population and thereby accelerating the detoxification process. But what's truly fascinating is the advent of genetically engineered microorganisms designed specifically for bioremediation tasks. Scientists have harnessed the power of genetic engineering to enhance the mitotic rates of these organisms and tailor their metabolic pathways to target specific contaminants more effectively. Consider the case of oil spills, devastating events that wreak havoc on marine ecosystems. Traditional cleanup methods are often invasive and ineffective. Enter genetically engineered bacteria designed to thrive in oil-polluted waters. These bacteria, through accelerated mitotic processes, multiply rapidly, breaking down the oil into non-toxic substances. This application not only demonstrates the power of combining mitosis with genetic engineering, but also represents a leap forward in our approach to environmental conservation. The implications of this innovative approach extend far beyond the immediate cleanup efforts. By restoring contaminated environments, we are also protecting biodiversity, safeguarding public health, and preserving natural resources for future generations. As we continue to face environmental challenges, the role of mitosis in bioremediation serves as a beacon of hope and a testament to the intersection of biology and environmental science. Thanks for your listening, and now comes to my conclusion of the presentation. The real-world connection between mitosis and bioremediation exemplifies the incredible impact of cellular processes on global environmental health. It's a vivid reminder that the study of life at the microscopic level can lead to monumental solutions for our planet's most pressing issues. I am immensely grateful to have had the opportunity to delve into such a captivating topic, and I extend my deepest appreciation to my family, the Kunshan University, my instructors, and peers for their unwavering support and encouragement.